From Microbe TV, this is Matters Microbial, a podcast about the wonders of microbiology, microbiologists, and microbial centrism. This episode was recorded on May 22nd, 2025. Hello, Micronauts, and welcome to our Quality Quorum for today. I'm your host, Dr. Mark Martin, Associate Professor of Biology at the University of Puget Sound in Tacoma, Washington. Today is the 95th episode of Matters Microbial. We're inching toward episode 100 ever so slowly, and we are 3D printing glowing pins in anticipation of this event, as you can see. I am so very grateful for every listen, view, and email. Truly. As I gear up to teach microbiology in the fall here at the University of Puget Sound, I have many microbially inspirational tools at my disposal. So I obtained this trophy for myself to remind me it is certainly not my first microbial rodeo. Thus, I intend to proselytize the microbial truth to the students with every bit of ATP hydrolysis in my body. I have long been very interested in symbiosis since middle school. The genetic and biochemical handshakes between the partners are fascinating to me, which brings me to Dr. Tom Miller's famous quote, as you can see, "All organisms are nothing but a bag of other organisms walking around." Really gives you an interesting perspective. One topic we'll discuss today revolves around aphids, which are a costly pest on many plants, including ones of agricultural importance. And there are wonderful stories about various microbial symbioses with aphids. I'll put some references to them in the show notes if you're interested. Another topic for today will be siderophores, molecules that help scavenge valuable iron from many microbes. Some of these siderophores fluoresce under ultraviolet light, and I often see them in the water bottle buddies isolated from student water bottles, as seen here. Which led to one of my microbiology students to come up with a painful pun about holiday cider and siderophores, as seen here. So, I'm delighted to welcome Dr. Tory Hendry, Associate Professor of Microbiology at Cornell University, to talk about the work her research team is carrying out exploring symbiosis, aphids, and fluorescent bacteria. Welcome to the Quality Quorum, Tory. It's wonderful to meet you.
Nice to meet you too.
We have lots to talk about. And can I just take a moment, and I want to say first off, congratulations on earning tenure. It's a big step.
Thank you.
What did you do to celebrate?
That's a good question. I feel like it hasn't fully kind of sunk in yet, so I haven't had a big celebration. We had a little bit of a party with some of the assistant professors in the department, which was really nice.
It is really nice. You have quite an interesting department. I've spoken to Esther Angert, for example, who I'm a longtime fan of. So anyway, I'm so pleased for you that whenever I hear these things, it makes me very happy because sometimes there's not a lot of positivity in the day.
That's true. You definitely have to celebrate everything good that happens.
Before we get started, I wanted to make something clear to listeners and viewers, and it's the role of iron in pretty much everything that lives. And most of us are used to the idea of iron that we use in hemoglobin. Why is iron important for microbes?
Yeah, so iron is an important cofactor in a lot of microbial processes. It's necessary for a lot of enzymes to work. It's basically used in everything. But part of the problem with iron is that it's mostly not biologically accessible in the environment. And so most microbes can't use most of the iron that's available. And so they've come up with a lot of different mechanisms actually to get iron from the environment and use it in a biologically relevant way.
And this is where I get into trouble sometimes, Tory, is that I'll think it's an adaptation to oligotrophic conditions, very low in nutrition and cofactors, which brings us back to water bottles. But it's also part of competition. Our own bodies tie up bioavailable iron in the bloodstream to keep bacteria from growing there.
Absolutely, yeah. So siderophores, in particular, ways to scavenge iron can often be virulence factors. So the microbial approach to kind of get over that host limitation of iron.
Yeah, it's absolutely true. And the second thing is that I know many people think of aphids as being pests on their house plants, but this is
Yep.
commercially important as well.
Yeah, aphids are one of the biggest agricultural pests in terms of vectors of plant diseases. So about 50% of plant viruses that are vectored by insects are actually vectored by viruses or by aphids. Aphids are essentially mosquitoes for plants. So they're sap-sucking insects. And because of the way they feed, they're really good at picking up viruses on plants and basically injecting them into new plants. And so they can be really
It's a
damaging, partly because they're feeding on crops, but mostly because they're moving pathogens around that can be very costly.
kind of like hopping hypodermic needles.
Exactly. Yep.
So you might as well just lay out then now. First off, I want people to know I'm going to put a link to your laboratory website for
Mhm.
your laboratory group in the show notes, and you do lots of interesting stuff. And I am biting my lips so hard I'm bleeding because bioluminescence and anglerfish has always fascinated me. But that's not what we're here to talk about. We're talking about the other end of your research.
Yeah, so I would say my first love in microbiology was probably symbiosis. I fell in love with microbes because of their diversity and kind of the metabolic diversity that they have that allows them to do so many things. And the way that eukaryotic hosts can kind of benefit from that and evolve with microbes to form partnerships really struck me. So I started in symbiosis, and that's actually how I learned about aphids, because there's a huge amount of really beautiful work looking at the way that aphids can form symbioses with intracellular bacteria. And so this is a very tight partnership, right? So aphids are sap sucking and they feed on plant sap because it's really high in sugar, but it's really low in other nutrients, especially nitrogen sources. And so basically, they need bacteria in order to synthesize amino acids that they don't have access to in their diet. And so this microbial partnership with bacteria has allowed aphids to expand into a new habitat to make use of plant sap, which is super rich in sugar, but hard to actually use and practice. So there's this like beautiful story about the way that aphids have all of these different tight partnerships with microbes that do things for them, and I loved that, but I ended up kind of getting interested in the microbial ecology side of interactions. And although there's really cool evolution that happens with symbionts that kind of get stuck with their host, their ecology kind of becomes that of the host. Whereas bacteria, of course, in the environment are very diverse, and they're having all sorts of ecological interactions. And so I was thinking a lot about how bacteria in the environment might be actually interacting with hosts. And this is something particularly for aphids, and I think, for a lot of things, that we're constantly interacting with a diversity of bacteria, and we don't actually know how or if any of those are impacting us. And so I ended up with in a lab that was studying Pseudomonas, and there had been kind of one paper that had come out showing that Pseudomonas can infect aphids. So Pseudomonas is really common on plants. It's a natural part of plant microbiomes. It's kind of always there. There's a lot of diverse Pseudomonas on plants. And so they're constantly interacting with aphids. And so I liked that kind of taking a look at, okay, well, these two things are coming into contact with each other, what happens when they do. And it turned out that when aphids became infected with Pseudomonas on plants, depending on the strain, they could die really quickly. So I have a figure showing that Pseudomonas can kill aphids. You can see that this varies across different strains. that can vary from, depending on the strain, as few as 30% of aphids that are dying in a few days, but up over 80% of aphids can actually die within just a few days from an infection. So these are really common bacteria that are on plants, so basically on the aphid's food. And when they encounter them, they can get infected at a pretty high rate. And once that infection happens, it seems that mostly it depends on whether the bacteria can grow inside the aphid or not. So on the left, you can see an image of an aphid that is infected with a Pseudomonas. So the Pseudomonas is naturally fluorescent. So that's a common trait of Pseudomonas that, as you said, comes from their siderophores. So this particular strain is producing the siderophore pyoverdin, and we have infected the aphid orally with the bacteria. The bacteria have grown to a really high density, so they can get up to 10 to the 7, 10 to the 8 cells per aphid. And so aphids are really small, so they're really just like filled with bacteria. And once they reach a certain density, they start to produce pyoverdin, and you can actually see that fluorescence. So here we're shining a UV light on the aphid, so you can see that fluorescence through the aphid's body. So this aphid is actually still alive, but she won't be for very long. Basically, once it gets to this point and she's just filled with bacteria, she's going to die. And so this is one of these virulent strains that we found on plants. And so after screening a lot of different Pseudomonas, we found that this can vary a lot, but it's pretty common to find strains that are extremely virulent on plants that must be interacting with aphids pretty regularly.
So for listeners and viewers, just a couple of things, if you don't mind, Tory.
Mm-hmm.
And the first is the difference between pathogenicity, the ability to cause disease, and virulence. And the way that I define it, and feel free to correct me, is the number of organisms it takes to cause disease causation. Mm-hmm. So you don't have to agree with it, but I like to teach it that way to students. Yeah. Just a couple of cells of Bacillus anthracis can cause anthrax, but it takes about 1 million of Salmonella to give you food poisoning. So they're both pathogens in that sense, but one is more virulent than the other.
Yeah.
I was also fascinated by the fact that the aphids themselves are fluorescent when they have a fluorescent pseudomonad. Mm-hmm. I'm going to say infecting them. Did you say that they're becoming intracellular?
No, so we're not entirely sure. We know that they're in the gut because they they're leaving the aphid in waste. So they're secreted in honeydew. Aphids produce, it's a very poetic way to describe the waste of aphids because it's basically sugar water, because they eat so much plant sap that they're excreting excess sugar that they can't use. So their poop is essentially sugar. And so there's a lot of bacteria in it. So we know that they're in the intestinal tract. It's not clear if they leave the gut to go into other tissues or not.
It's fascinating to me because, of course, some ants will actually farm aphids for that honeydew. I've talked to classes many times about honeydew and what a silly expression that is. But if you think about it, I mean, there are many organisms that don't fully digest their food. In fact, cows re-eat the waste products. Yep. And this allows stuff to be transferred to the next generation as well.
Yeah.
So have you ever, I mean, if these are actually visibly UV, can you just shine a UV light on aphids in like in the wild and see that they're naturally occurring as well, or?
Oh, that's a really good question. We haven't tried that in the wild. We do use it in the lab to pick aphids that are heavily infected if we're using that for experiments. And this has been it brings up a challenge that we've had. So it's easy to do these experiments in the lab. It's a lot harder to do things in the field. But part of the problem is because infected aphids die so quickly. You typically, if you find living aphids, they're healthy. They're not infected. And interesting kind of aspect of aphids is that most aphids don't actually have much of a gut microbiome. They have these really tight interactions with intracellular bacteria, but they don't typically have a lot of bacteria in their gut. And so if you find something like this in the wild, I would assume this would be an aphid that would be dying near death. And so finding that stage when they're heavily infected and right before they die is a challenge that we haven't really figured out how to deal with yet.
May I also ask if pyoverdin synthesis is under quorum sensing control?
It is in some Pseudomonads. It's not in the ones that we're typically using, but that does happen.
So once again, this is important for folks to understand when they take microbiology.
Mm-hmm.
There are as many rules as there are microbes, it seems, some days.
Absolutely.
So just as you, and there are certain types of molecules that people say are under quorum sensing rules most of the time, and some siderophores are, but that might only be in some members of that larger taxonomic group, if I understand correctly.
Yeah. Yeah, so we have been looking at kind of the diversity of Pseudomonas on plants and how they interact with aphids, and the fact that they're can be so virulent and are so common led us to kind of start thinking about well, what's happening to aphids in the field? Are they just constantly being infected with Pseudomonas and dying, or do they have some kind of way of dealing with this pathogen? And part of the way that aphids kind of deal with anything is one of their adaptations is they reproduce at a very high rate. So essentially, aphids are asexual. They're parthenogenetic, so they can make clones of themselves. Most aphids that you see out in the world are female. They're typically described as being born pregnant because when they're born, they're already making the aphid embryos that they'll give birth to. In about 9 to 10 days, they become adults, and then they start reproducing, and an adult female aphid can make 5 clones of herself a day. So a single female aphid can really just like take over a plant pretty quickly. And so this is something they do really well. And it seems that they invest in reproduction potentially over immune investment. So the P aphid genome in particular showed that they have a reduction in some immunity pathways compared to other insects that we understand, like Drosophila. And they seem to mount kind of less of an immune response than you would expect to pathogens. And so maybe they don't really care actually that much about pathogens the pathogens don't have very much evolutionary selective pressure on them because they can just kind of reproduce regardless. But we had kind of noticed something by chance in the lab, which is that we seem to have a hard time infecting aphids with some of our more virulent strains. And it seemed as though they didn't really want to eat. So aphids normally eat all the time. You know, once they settle, they just start feeding on plant sap, and they're constantly eating. And we do a lot of infection assays and kind of an artificial plant sap that we can put bacteria in and infect aphids orally, so that it's controlled, but it's more kind of natural. And it seemed like we were having a hard time getting them to actually want to eat with some of the strains. And so we thought, well, maybe they're actually avoiding the bacteria because they're pathogens. And so we tested this. And this was actually the first undergrad project that I had in my lab shortly after I started. So my first REU student joined the lab, and she figured out how to kind of set up choice experiments where she was giving aphids a choice between leaves that had been treated with bacteria or a control treatment. So they were just treated with a buffer. Just to show that there could be some kind of differentiation between those different treatments. And we found that aphids could avoid bacteria. So they were showing, depending on the strain, up to 70-80% of aphids were choosing the control treatment over leaves that had bacteria on them. And so we wondered if maybe this was actually an adaptation where they were avoiding, virulent bacteria. And so initially we tested, I think, 8 strains in our first paper, and that showed that they didn't actually avoid all bacteria. So I thought that maybe they just don't like bacteria. I don't like bacteria in my food either. They're smelly, whatever. There's something about it that they don't like. But some things they don't actually care about. There's only certain strains that they seem to mind. And so in that paper, we found that actually the more virulent strains were the ones that were actually avoided by aphids. So they seemed to be able to tell that there were bacteria there and also discriminate between different bacterial strains to show, to figure out which ones were the ones that they should avoid and which ones they shouldn't. So we worked through this to figure out what was actually happening by first thinking about what we know about Pseudomonas and what we know about aphids. Of course, one of the common features of Pseudomonas is that they are fluorescent. So I really like this image of fluorescent Pseudomonas that's showing a plate that we took a leaf and we made a leaf print. So we just took a leaf, put it on a plate, an agar plate, took the leaf away and incubated it. And so you can see that there's a diversity of different things that have grown, some fungus and different bacterial colonies in the middle. But when you take this leaf and image it under UV light on the right, you can see that some of those colonies are fluorescent. And so this is like a really distinctive, cool feature about Pseudomonas, of course, is that they glow. And so this glow is really well known. And another thing that's really well known about aphids is that they use color vision a lot when they're trying to find plants. So there's all these cool papers from like the 70s that look at what colors aphids prefer. So people often use those yellow sticky traps to kind of collect pests, and those work because aphids are actually attracted to the color yellow. So there's a lot of work trying to figure out why that is. I remember reading one paper where somebody had put out different colors of construction paper and just looked to see which paper the aphids walked to see which colors they liked.
I love it.
Yeah, no, it was fun. But what we know from that is that aphids are using color to find plants, particularly at a distance. So they're not really strong flyers. They're kind of taken around by the wind, but they need to be attracted to plants, and so they like green. They also like yellow. They actually don't like blue, right? So they're trying to avoid presumably the sky because they don't want to just float away. And so we wondered whether Pseudomonas could actually be, because of this fluorescence, whether they could actually be changing the color of light on plants in a way that aphids could detect. And so we tested this in a few different ways. In one treatment, we took plants and we took leaves and we put different bacteria on them. And so we used bacteria that we knew were avoided, some that we knew weren't avoided. And we also had a deletion mutant that is not fluorescent. So the wild type of the strain produces a really strong fluorescence. It's actually the one that I was showing pictures of before. And the knockout is not fluorescent at all, so it doesn't produce pyoverdin. And so you can see in the image here where we are taking fluorescent bacteria on leaves when we're measuring the color of those leaves using a spectrophotometer. When you look across different wavelengths, the different leaves treated differently. So avoided strains that aren't avoided, or control treatments that just are buffer, or a leaf by itself. The leaves are mostly the same color across wavelengths, but where they vary is in 425 to about 450, and that's the blue range of wavelengths of visible light. It also happens to overlap with the fluorescence emission of pyoverdin. So pyoverdin absorbs UV light and then emits it back out as visible light in the blue spectrum. And so we found that the strains that were avoided were actually making the leaves more blue than strains that weren't avoided. And it seemed the aphids could detect this difference and were avoiding those strains.
You know, I have to say that I am adoring this work, and I really appreciate your clear explanations of it. There's a particular paper I introduced to my students. I'll put a link to a blog post I wrote about it in the show notes, having to do with hoverflies and aphids.
Yeah.
And the shorthand is the hoverflies are attracted to a particular metabolite of a bacterium of the aphid, that semiochemical. It's called a pheromone. And to me, you know, I had my mind in the idea like maybe the aphids are smelling. And what I like about that is you go back to first principles and you say, what is it that they're trying to perceive? And they have color vision, and that this makes perfect sense. The other thing that I'm fascinated by is the idea of bacteria that live on the surface of leaves, the phyloplane, and how that can, you know, what advantages that they have. And obviously, there are things being exuded by the plant all the time that can be used for metabolism. And so that's a particular niche that can be exploited by the bacteria. And then here you have the other side of it, where the aphids can actually evaluate the health of the plant. And, well, not the health of the plant, but whether or not there's the Pseudomonas on it. I don't know that necessarily the Pseudomonas has a negative effect on the plant.
Yeah, no, I love that hoverfly story. That's really cool. And I will say that when we first started thinking about this, I assumed that it would be odor based, right? That they were smelling
Yeah.
the bacteria. I didn't really the visual aspect really seemed like a long shot to me. But the main reason that we went down kind of the visual rabbit hole was actually that it was a little bit easier. So you can knock out odor reception in insects. They're like blockers that you can buy. But it was a little bit unclear to me how I would be able to tell if the aphids couldn't smell, like what would be a positive kind of control for that. And so that experiment seemed hard to do. But the choice experiments, the visual choice experiments, were easy to do because we could just do those experiments in the dark. And so that was actually the first thing that we did. We did the choice experiments. So we had shown that aphids could avoid the bacteria. We did the same things in the dark. They still found the leaves. It took them a little bit longer because they didn't have eyesight, but their preference went away. So there was a clear visual component that they were relying on there.
It's amazing. And this is a project an undergraduate championed originally, and that's
Yeah,
important for folks to understand.
absolutely.
No, I run into students all the time that say, I'd love to do research, but I don't know enough.
Mm-hmm.
And it just breaks my heart because, of course, you don't. This is the most expensive piece of equipment in the lab.
Yep.
You know, the brain and the eyes.
Yep. Absolutely. So we found that this was pyridan based. We're using the diversity of strains, in particular a strain that wasn't producing pyridan. So they needed pyridan in order to avoid the bacteria. And it seemed that the more virulent strains were actually those that were being avoided, and so this would be potentially adaptive. It wasn't. We did test the pyridan deficient mutant to see if it was less virulent, like maybe they're actually avoiding the thing that is a virulence factor, and that didn't seem to matter. But there is a correlation between kind of how well the strains grow on plants, what density they reach, and then how virulent they are to aphids. And so we thought that maybe producing pyridan was an adaptation to growth on the leaf, and then that was therefore a cue that this bacteria would be abundant, and therefore you should avoid it. So that was what we kind of initially thought, but we were looking at kind of a limited diversity of Pseudomonas. We just had 8 strains, and they were mostly from the kind of species complex called Pseudomonas syringae, which is really well studied as a plant pathogen. And so you mentioned that there's also the plant interaction. Many of these strains are plant pathogens, but most of them aren't. And so we had kind of a mix of plant pathogens and non-pathogens in that first experiment. We were concerned that maybe actually what the aphids were paying attention to was plant-mediated, so the pathogenic bacteria on the plant is having a response, and herbivorous insects can detect that, and so they may or may not prefer a plant that's actually infected because it's easier. But we were able to get the same responses without kind of a plant effect, even without plants being present, just like bacteria in artificial diet cause a behavioral response. So we did think it was the fluorescence, not the kind of the plant interaction. But in order to get a better sense of whether this was something that was kind of more ecologically relevant, true across more bacteria, we wanted to expand our Pseudomonas collection. So I had a grad student who went out into the field and we sampled leaves from pea plants. So we're working with pea aphids for the most part. And so we're looking at pea plants on farms. And we sampled bacteria on leaves, and then we just used UV light to find fluorescent pseudomonads on plants or on plates. And so what we found was actually a huge diversity of fluorescent colors. So out of a collection of about 50 something different unique strains, so these were unique in terms of their 16S sequence, we found a pretty wide range of different bacterial fluorescence colors. So I have some example plates on the left here where I'm showing streak plates of different strains, and you can see that they vary in color from a pretty blue fluorescence to actually quite green. They're beautiful. There's also a huge amount of variety. And so my student wanted to kind kind of catalog this in a more controlled way. So she took these strains, she made spread plates, so there's just bacteria evenly spread on the plate, and then she imaged those under UV light and then used ImageJ to calculate the color of the fluorescence. And here we're plotting that color in terms of how green versus how blue the fluorescence is. So it varies a lot in how intense fluorescence is. So on the lower, left of the graph would be strains that are not fluorescent at all. There's basically no light intensity. But on the upper right, those are extremely brightly fluorescent. And then it also varies in kind of how green versus how blue the bacterial fluorescence is. So we we're curious about this because we had shown that blue fluorescence was avoided, but we didn't know if that would be true kind of across different strains or whether it matters what the color was. And so my student took a subset of these strains and did choice experiments again to see how the aphids would react to the different fluorescence. So it was a similar kind of choice setup. We changed it a little bit. We actually treated whole plants with the bacteria versus control treatment. And then we also, initially we had used aphid nymphs because they're easy to work with. They kind of crawl around on plants. But in this experiment, we actually used winged aphids. So these would be the aphids that are dispersing. So when a plant gets really crowded, individuals start to produce offspring that have wings. Those are called alates. And those alates, they really want to fly. They need to fly to other plants. They'll fly and then they'll immediately, once they find something, a plant that they like, that they want to settle on, they'll immediately start reproducing and they'll make lots of babies and kind of crowd the plant again. But that's, in a lot of ways, it's kind of the really ecologically important step in kind of aphid ecology, because those are the aphids that are moving to new plants, taking viruses with them, and kind of adding that additional infection from the plant point of view. So we had plants that were spread out in a tent, and they were treated with either bacteria or control. And then we had to release aphids so that they would have to fly to choose among the plants. So we basically hung petri dishes from the top of the tent. My grad student called these flying saucers. So the aphids were put in the petri dish, and they had to fly, and they could choose among the different plants. And we found, similar to our previous work, showing that some strains were avoided. Some of our new strains were similarly avoided. So we are showing here, this is the graph on the left showing aphid behavior is a little bit different in that it's showing the percent of aphids on control plants. So basically, if aphids have no preference for control versus bacteria, you would get 50-50. So you should have 50% of aphids on your control plants. But if you get more than 50%, they're showing a avoidance of the bacteria. So that we can see a lot of the strains, like the one in pink in particular, are highly avoided, and you're getting 75% of aphids choosing the control plants instead of the bacterial plants. So that was consistent with what we'd found before. But what was really striking is the strains that I have boxed here in red were actually preferred by aphids. So in the case of the kind of the teal green strain, again, like 75% of aphids were actually choosing the plants with the bacteria compared to the plants without the bacteria. So this completely kind of changed what we were thinking. Because we had been thinking that maybe this was an adaptive behavior, but maybe it isn't. So what was interesting about these two strains that are attractive is that, at least in culture, they seem to be more green in their fluorescence. So they're producing green fluorescence in culture. We haven't looked to see that that's what they're doing on leaves, but potentially they're actually making those leaves more green. And because we know that aphids are attracted to green and avoid blue, maybe what's happening is that the bacteria are actually kind of eliciting a behavioral response that's driven by a sensory bias. So it's this underlying sensory bias of color preference that the bacteria are kind of causing that isn't necessarily adaptive from the aphid point of view. And so that was really interesting and kind of expanded our thinking about, well, how is this actually working in nature? What is happening to the aphids? How are they benefiting, or is this something that is just kind of happening to them and it isn't necessarily beneficial in terms of the interaction?
This is amazing stuff. I have a couple of questions
Mhm.
that I hope are occurring to listeners and viewers. The question is, well, first off, how do you put the bacteria on the plants? Do you spray using a mister?
Yeah, so typically we suspend the bacteria in buffer, and then we have like a fine mist sprayer that we spray on, and so it's meant to kind of create an even layer of bacteria on the plant, which isn't necessarily how they occur in nature, but actually bacteria on plants are typically often arriving in rain, actually, so it's not
Hmm.
that different than how they would normally kind of inoculate a plant.
I'm also wondering what the natural population of bacteria on leaves are. And what I mean by that is the numbers of them.
Yeah.
And I don't know if you've done, maybe you have done that work.
Yeah, so a lot of other people have done that work. And so it's obviously going to vary a lot from leaf to leaf and plant to plant and strain to strain. But strains that, Pseudomonas in particular, that can grow really well on plants can get pretty high densities. So people are often measuring like per leaf densities. So on a given leaf, depending on the strain, up to 10 to the 6 cells. So those are probably not kind of evenly spread the way we make them in lab, but they can reach pretty high density. So that was
Cool.
another kind of big question. Like we can get this to happen in the lab, but is this ecologically relevant? Do we actually see bacteria at this level in the field? And so we have done the experiments in the lab where we vary the density of bacteria, and you do reach a point where there's not enough bacteria that the aphids don't care. But for some strains, the population size that we're getting is like 10 to the 3, 10 to the 4 cells, and aphids still respond to that. I
Mm.
think that it doesn't necessarily take a lot of bacteria for this to happen.
This is just amazing to me because we don't think about, people get unhappy with when I say the way that an organism perceives.
Mm-hmm.
And that includes what plants perceive.
Mm-hmm.
It doesn't mean think about. So
Yeah, yeah.
no angry emails about that. Um, or what bacteria perceive or what aphids perceive. And it's a subject that is so fraught to bias the way that we see and perceive the world. And what I like about this is you take a step back and just here are the possibilities. For example, when you put things in the dark to see if the choice experiments work,
Mhm.
which I think is really lovely.
Thank you. It was easy to do. Yeah, so what we're following up on now is a couple of different things. One, from the aphid point of view, we're interested in the attraction angle. So because we had initially hypothesized that this could be adaptive, the aphids are avoiding pathogens, but we didn't know if these particular strains were pathogenic or not. So potentially they weren't pathogenic. Maybe there's actually something good about them because of the way they interact with the plant or something. And so I
Mhm.
have a current grad student that's been following up on this, and it turns out that those attractive strains are very pathogenic. So most of these new strains that we have collected on plants are actually more virulent than any of the ones that we looked at previously. So they're growing on plants to really high density, and they are potentially quite damaging to the aphids. So then you have a situation where the bacteria are causing the aphids to potentially go to plants where they would survive less well. So we're testing whether this could kind of create an ecological trap. So this is a term that ecologists use for when organisms are attracted to a habitat where they actually don't have very high fitness. And mostly it occurs in cases where there's been some kind of anthropogenic change to a habitat that again is kind of eliciting a sensory bias where causing the animal to make a bad choice. So a good example would be mayflies. They're attracted to polarized light, which they're using to find water. But asphalt also reflects polarized light, and so mayflies can be attracted to roads, which is obviously not a good habitat. So whether the kind of sensory bias, the preference for green, could actually be causing aphids to choose plants where they ultimately have lower fitness is something that we're currently working on.
It's like stop and go signs.
Mm-hmm. Absolutely, yeah. And then the other thing that we've been looking at is more from the bacterial side. One thing that we discovered when we were doing these experiments is that a lot of these. So initially, our strains that we were using, if they were avoided, they were pretty consistently avoided. So every replicate that we did, the aphids didn't like them. With a lot of these new strains, sometimes they were avoided, and sometimes they were actually preferred, or sometimes the aphids didn't care, and there was a huge amount of variability across different replicate experiments. So imagine my grad student, she was doing these experiments actually during COVID when we first got approval to go back into the lab after lockdown, but she's in here by herself because nobody else can be around. And so she's doing experiments where every time she does them, she gets a different outcome. So this was a real, like, am I even staying in science kind of moment. But we worked through it. We essentially did this several different times. And we think that this is like a real thing. It's not just, you always get experimental variability. But for some strains, they seem to really distinctly switch, where sometimes they're attractive and sometimes they're avoided. And we think it might be that they're actually changing what siderophores, or whether they're producing siderophores under different circumstances. So one of the things that I think is really cool about siderophores is that especially Pseudomonas, but a lot of bacteria will actually have a lot of them. And they're essentially all doing the same thing. They're excreted into the environment, they scavenge iron and allow the bacteria to take it back up. And so why, particularly in bacterial genomes that are very kind of streamlined, they're just doing what the microbes need, why have so many different types of siderophores has been a question that's really been kind of driving me. And so there is some suggestion in literature that different siderophores are turned on under different environmental conditions. So it might be temperature or different nutrient availability or pH. Basically, they may work, one or the other may work better under different circumstances. And so there might be something about the way we're doing the experiments on plants where there's enough kind of environmental variability that they're changing which siderophores they produce. And so one of the strains that we know switches a fair amount, we can see it in different culture conditions. Like sometimes it's very green and sometimes it's very blue. So we know that it has this ability to kind of change color. And we think that might be happening on plants. And so that causes the different kind of behavioral responses. So we're interested in trying to figure out, like, what are the different environmental conditions that drive that? How often does this actually happen on plants? And then how does this impact the aphid behavior?
So this is really interesting to me because when I isolate bacteria from nature, I often see really variable phenotypes for anything that I happen to be studying.
Yep.
And then with time and culture, often that variability is lost as they actually evolve to fit laboratory conditions. And what I really like is you're using funny expression, you're using undomesticated bacteria for this. And I love the fact that we talk a lot about biosensors, and if in a way that's what these bacteria are doing is they're sensing something, changing the siderophore, and that change is being perceived by the aphids.
Yeah. So, I mean, I think like it's not clear to me that there's any sort of benefit to the bacteria from the aphid interaction. They're maybe just doing whatever they're doing, adapting plastically to their environment. But then that has these like kind of multi-trophic effects on things that are, eating plants, I think is, it's really complex, but really interesting to me.
Fascinating stuff. What occurs to me is, do you think that this could be used to reduce infestations by aphids on plants?
Yeah, so that's definitely something we're pursuing. We do have a patent for using fluorescent bacteria to control aphids. My interests are really fundamental kind of bacterial evolution and ecology, but like the applications of this are really really interesting. So we have played around with using fluorescent bacteria as deterrents. And people are doing a lot of really cool stuff with plant microbiomes, like using plant beneficial microbiomes to improve plant growth. We know that a lot of plant microbiomes can be protective against pathogens, so beneficial kind of commensal members of the microbiome, just like in our microbiome, right, can prevent colonization by plant pathogens. And so whether we could also kind of design and engineer a microbiome that was beneficial to plant growth, could kind of control pathogens from colonizing and also deter aphids is something that we're also playing around with. The potential for the the both attraction and deterrence to be used is also there, because sometimes people will use kind of what's called push-pull pest management, where you have like a trap plant that you're attracting your insects to, your
Hmm.
pests, and so that's just a throwaway. It's where they all go, and then something that is a deterrent on the other plants, and so. People will use this in greenhouses sometimes because aphids can be really hard to control in greenhouses. And so basically, if you have a trap that kind of lures the aphids there, you can protect the other plants. So I think there's a lot that could potentially be done, and we're just trying to figure out kind of the basic biology in order to get to that point.
My mind continues to reel over the issue of the color being sensed, the color that's being reflected off the leaf by the aphids. And it makes me wonder things like, if you filter UV out of sunlight, do they still feel attracted? Is it the color per se, or is there some spectral mix? Can you just shine a primary color on a leaf or a target and get the same kind of effects?
Yeah, those are really good questions. So, some of the experiments that we did when we were trying to figure out how everything worked was that we did actually block UV. So, we were doing our choice experiments under plastic boxes, basically. So, we have our full spectrum light shining down. This is in the lab, so it's kind of full spectrum lights. Those actually don't go that far into the UV. So, we add supplemental UV that we've shown mimics sunlight pretty well. It doesn't go super far down, but it seems to work well
Mhm.
enough. And so we would do choice experiments under boxes that were made of UV blocking acrylic. You can buy this because like museums use it to keep UV from kind of destroying paintings and stuff. And then acrylic that transmits UV. And so without UV light, the aphids don't show any behavioral difference. They aren't able to perceive the bacteria. They need UV light to be there. So that was our first kind of indication that this was, so we knew it was visual at that point, and that it was also kind of UV mediated. But one like kind of additional question there is, because aphids do see UV light, whether they were actually detecting differences in UV light. So we've shown that when you have bacteria on plants that are producing siderophores, they're absorbing that UV light, and so there's much less reflected UV light coming off the leaves. And so potentially aphids could actually be perceiving that instead of the blue. So we've sort of tested this by looking across different bacteria that vary in the color and also how much UV light they're absorbing. And there doesn't seem to be a correlation with the UV absorbance and behavior. So we think that it is really the color and not the UV absorbance that they're paying attention to. Which
I
suggests
ha-
that maybe just color on its own would actually be enough. So, as I said, people have done experiments with just different colored pieces of paper. And so we have thought a lot about,
Mhm.
like, do you need to have bacteria there? Could you just shine colored lights? And that potentially would work pretty well, like in greenhouses where you can control the lighting pretty well. We haven't tested that, but I think that that has a lot of promise. It would, potentially be less useful in the field, and so that's why I think the kind of controlling a plant microbiome might be more useful. But there's definitely, I think, something to look at there that we need to follow up on.
I think it's lovely work, and what I appreciate it, and I want to be very clear on this, is there's a level of it for even the most novice student,
Hmm.
as well as the most advanced postdoc,
Yeah.
and that's kind of rare to see that level in one lab.
Yeah, I love it. I mean, I think the aphid experiments are great because they are a really good way to as an entry point for undergrads. It's like something
Mm.
that is a more accessible sometimes than microbiology can be. That's also like the fluorescent bacteria. The fact that you can see them and they have this cool phenotype is really compelling. But the aphid experiments, we can design pretty simple experiments that can be done, in just a few days, and so that can be a really nice way to particularly for summer projects for undergrads to kind of get them involved.
It sounds like a little bit of changing gears, but I want to throw something out to see what you think. I have a friend who many years ago was looking at a species of island gecko that was sexual and one that was asexual.
Mm.
So this one, the asexual one, they were essentially clonal, and she was able to show a higher parasite load in the asexual
Mhm.
ones.
Mhm.
And I think this has been seen in other systems. And so whenever I hear about a parthenogenetic or asexual species, I always wonder about what kind of loads of,
Yeah.
I hate to say the word pathogenic bacteria, but what kinds of sensitivities they have.
Yeah.
Do you think any of this is relevant?
Yeah, definitely. So, I mean, people have looked into that a lot in terms of like the benefit of sexual reproduction because it is in some ways more difficult than asexual reproduction, right? With aphids, because they are reproducing so quickly and they often reproduce asexually, you tend to get a lot of genetic drift. Different aphid clonal lines can be very different genetically
Yeah.
and phenotypically. And so we may very well see differences across different aphid clones. I mean, people tend to in terms of phenotype. We haven't really looked at that level of diversity. We might see, that some clones are more affected than others. We do find this kind of similar effects in terms of the susceptibility to Pseudomonas pathogens and also behavior across different aphid species. So I think the kind of general principles are probably true across aphids, although we have found some species that don't seem to care actually. But that would definitely be something to look into, like what the genetic diversity of a genetic variation in the aphids kind of does to the interaction.
What I love about science is many times people will, especially in the current climate, they'll say this particular type of research is silly or useless or whatever. Just bear with me for a moment. And then we have tons of examples where we didn't understand how applicable something is. And there are tons and tons of examples. And I just want to alert the viewers and listeners to this concept that you really can't make judgments on the value of research. Historically, we know that the research that we think doesn't apply to the human condition. For example, organisms that live in thermal pools in Yellowstone that made PCR as we know it possible.
Yeah.
So I wanted to just beat the drum for how important science is regardless.
Absolutely. I think, the benefits of fundamental scientific research are huge and hard to predict. And so that's why we need to understand how everything works in order to make any sort of advancements.
And the value of your research is that most all of us have had to deal with aphids at one point or another.
Mm-hmm.
And so go ahead.
Yeah, so I mean, people always, the first thing when you say aphids, like, oh yes, I couldn't get them off my plants. I mean, in addition to being agricultural crops that are problematic, they're also actually really hard to control. So part of that being the fact that they reproduce so quickly, they can also develop resistance to a lot of pesticides. But the typical way that aphids are dealt with in some kind of crop systems is with pesticide use, right? So prophylactically, crops would be sprayed with chemical pesticides before whatever aphid they're susceptible to would tend to be choosing host plants. And that's not great in terms of environmental sustainability to just be spraying pesticides broadly. So there's a lot of interest from growers and from individuals in being able to use more kind of sustainable methods. And so that's what I like about being able to kind of harness bacterial diversity, naturally occurring microbes that are on plants anyway, that are part of plant microbiomes, that could be kind of used more for our benefit to control where aphids go.
I'm going to put a link in the show notes to microbial leaf diversity because I think that will be fun for people to think about.
Mm-hmm.
I spend a lot of my time looking at individual items and thinking, what's the microbiome associated with it? You know what I mean? Yeah. So this is very important. I really think that what you've described to people is applicable and interesting, and I would also argue a lot of what you've discussed is fundamental and that it's really accessible to people. And I appreciate that very much.
Thanks.
Do you think you might spend a minute or two talking about your path to the microbial sciences? And I want to explain why I'm asking. Is I get students all the time that think there's only one way to become, for example, a microbiologist. And if nothing else, this podcast has shown a variety of paths. What's yours?
Yeah, I think it's a great question. I do think it's really important for students to see. I come from a really rural background. I was a first-generation college student, and so the idea that you could do research as a job was something that was not at all clear to me until I went to college. And I think I feel very lucky to have gone to a school where research was really accessible. So I went to a small liberal arts school where it was very clear how you find research opportunities. There was a selection process. You could say what faculty you were interested in. You could end up working in their lab during the academic year of the summer. And so this is how I really fell in love with research. And it's also driven a lot of what I do now in really focusing on undergrad research programs and making sure that students have access to research opportunities. So I think that's really important. So I was drawn to ecology and evolution without kind of knowing why. I guess I really liked the way that type of research was explaining the diversity that we see in the world. Like why do things work the way they do? And I didn't fall in love with microbes until a little bit later. So I didn't take my first microbiology class until my senior year of undergrad, actually. And at that point, I'd mostly been kind of thinking of macroorganisms, plants and animals, and then thinking about bacteria and just that they could live everywhere and do anything was just astounding to me. So that kind of got me into microbiology, and I was thinking about that a lot. I knew I wanted to go into research, but the thing that I always like to kind of share about my path is that it was not always straightforward and didn't always work the way I wanted it to. So in undergrad, I applied to grad schools. I was mostly focused on animal behavior at the time, actually, because that's what my undergrad thesis had been in. I didn't get into any of the programs I applied to. I was completely out of success there. And I'm actually really glad about that because I think the way things worked out was the way that was best for me. But of course, at the time, it was demoralizing. But I didn't really know what I wanted to do. And so I ended up working in a lab for a couple of years as a research technician. I got a lot more experience and thought more about what I liked, what I was drawn to. And so I always encourage students to think about taking time off and thinking about what they want rather than just feeling like they need to go on a specific path. You can take time to figure that out. And so then I realized that these microbiology courses and things that I had experienced were really sticking with me, and I was really drawn in that direction. And so I ended up applying to microbiology programs. I ended up actually getting my PhD at University of Michigan in ecology and evolutionary biology. So it wasn't in microbiology at all. I was the only kind of weird microbiology grad student in the program, and that was interesting. But I think it was really, I learned a lot of fundamentals about ecology and evolution that have really been valuable to me. But because of that, I've also needed to kind of bounce around a lot in terms of getting experience. So I worked on bioluminescent symbiosis in grad school, which is, as I said, my first love. I always say that I have mostly worked on bacteria that glow. So within bioluminescent symbiosis, bacteria make light, so they're producing their own glow. Pseudomonas, of course, glow under UV and fluorescence, so I like to stick to things that glow. So I was working on a system of bioluminescent symbionts that are not culturable, so they've become really restricted to their hosts. And it was a super cool system, but we couldn't grow the bacteria, we couldn't keep the fish that easily. They're deep sea fish. It's like the least tractable system in the world. And so I wanted to get experience in my postdoc where I could actually do experiments and start to think more about bacterial ecology, because that's kind of where my interest had gone. And so I ended up looking at labs where I could study insect interactions, because I think when it comes to studying host microbe interactions, insects are where it's at. There's a huge amount of diversity among insects, and they have such cool interactions with microbes, and they're super tractable often. And so I ended up in the lab studying Pseudomonas that had an interest in kind of pursuing this interaction with aphids, because it had been shown that aphids could infect, the Pseudomonas could infect aphids. And I've just kind of stuck with that since then. So we work on a few different insect systems now, but I love that they're really tractable, and so we can We can look at the bacterial diversity in terms of phenotype and also genomes, and then we can do experiments to figure out how everything works, and kind of put things together in complicated ways within kind of pairwise interactions or whole community interactions, in order to figure things out. So that's been the system that I kind of developed in my postdoc and then have and stuck with into my faculty position. But like across those different positions, I was in animal behavior, I was in a plant evolution lab, I was in ecology and evolution department, a plant science department, an ecology kind of department. Now I'm in microbiology. So I sometimes don't know exactly how to describe myself. I guess I'm a microbiologist now, but I feel like all of these different experiences have really been beneficial in terms of getting me to where I am now and how I think about things. And so I always like to emphasize to students that when your path is kind of twisty, that can actually be a good thing rather than feeling like you don't know what you're doing. You're just kind of learning a lot.
You go to the cool questions.
Absolutely.
It's as simple as that.
Yep.
Thanks for sharing your path.
Yeah.
I wanted to, as we close out, I wanted to say I'm very grateful for your time. I was awfully enthusiastic about your work. I hope that was okay. Mm-hmm. But it touched on aspects of things that I see in my laboratory and in my classroom. So it was very interesting to me. So I want to thank you for that and the clarity of your presentation. And I also want to be sure to congratulate you again on earning tenure at Cornell. And I want to wish you and your laboratory group and friends and family the very best.
Thank you. It was a pleasure doing this.
Thanks so much. This has been Matters Microbial, a weekly podcast about the wonders of our microbial world and the people who study it. You can send questions, suggestions, or comments to me at mattersmicrobial@gmail.com. Show notes from today's episode with, as usual, epic links can be found at microbe.tv/mm. If you like our work, please consider supporting us at microbe.tv/contribute. I'm Doc Martin, and you can find me in the biology department of the University of Puget Sound in Tacoma, Washington. Dr. Tory Hendry can be found in the Department of Microbiology at Cornell University in Ithaca, New York. Many thanks, as always, to David Renata for superb editing and Reber Clark for the wonderfully quirky music that makes me smile every time I hear it. I hope that you've enjoyed being part of our Quality Quorum for today. See you next time on Matters Microbial.